in the base, where actually here it's a nitrogen, and in all of them actually. So at the position 1 in the bases, it's just simply 1 through 9 or 1 through 6. And here 1 prime, we're talking about the sugars, and that they are a deoxyribose. So the sugar is a 5 carbon sugar. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And of course it has an oxygen there. So it's a ribose sugar. And so let's see, the next obvious question is, we often describe DNA as a sugar, phosphate, and a base. Base, sugar, and a phosphate group. So that makes up the nucleotide. So this is a nucleotide when we have a ribose sugar, phosphate, and the ribose sugar, the base, and the phosphate group. So that's a nucleotide. What's a nucleoside? Doesn't have the phosphate group. So these will be nucleosides. And look at how the sugar and the phosphate are linked to each other. We have the sugar, have the phosphate, the sugar, phosphate, sugar. That's why we call it the sugar phosphate backbone. The base is over here. Of course, this looks better, but he just put an A here or G here and so on. Sugar phosphate group. So that's one side of DNA. The other side of DNA will have the same thing. So how do we describe DNA? We give it a definition. What do we call DNA? How do you describe it? What would be your description of DNA? It's a double-stranded helix, and the pairing is anti-parallel. So DNA is a double-stranded helix, although here I'm only showing one side. But obviously it has two sides in pair. Let's see if that's on the next one. Let me skip this one for a second. I guess they're not going to show it the way I want it. So it's a double-stranded helix. Adenine has 2 hydrogen bonds to thymine, and cytosine has 3 hydrogen bonds to guanine. And it's a helical formation. This diagram is how they determined that it was a helical formation using X-ray diffraction, allowing X-rays to go through the DNA, and the pattern that it forms revealed that it was a double-stranded structure, and that it was helical, and that there was pairing of the bases. So Rosalind Franklin is the one that actually was responsible for the X-ray diffraction part of Watson and Crick determining the structure of DNA. So this was just only one part of the scheme that was used. Of course, Watson and Crick put it together, saying that adenine has 2 bonds with thymine and so on. So a purine is always bonded to a pyrimidine. So in DNA, the ratio of purines to pyrimidine is always 1 in DNA. Because if you have one purine, it has to be bonded to the pyrimidine. So if you added up all the purines and all the pyrimidine, the ratio would be 1. That's not true for single-stranded DNA, and it's not true for single-stranded RNA, because they don't have to eat. It's one of the things that will tell you if a molecule is double-stranded or single-stranded, because double-stranded will be very, very close to 1. There's more to say. There were other people that Watson and Crick used, like Chargaff was actually a person that literally added up or determined all the purines and all the pyrimidines to see that the ratio in double-stranded DNA was 1. And so this is the actual pairing of the DNA, the two hydrogen bonds. It's unique here because hydrogen generally only bonds one time. But in DNA, hydrogen forms two bonds. A strong bond and a weak bond. Strong bond here, and then a weaker bond on this side. So hydrogen forms two bonds. Hydrogen forms two bonds in proteins as well, and also forms two bonds in water. But for DNA, hydrogen, so we got two hydrogen bonds for adenine and thymine, and three for guanine and cytosine. I want to say also, the figures in this diagram will not correspond to your book, but the figures are the same. This one, the figures, in your book, I think he does classical genetics first, and then goes to molecular genetics. And so in this book, which is the same author, he did molecular genetics first. So you see in chapter 2, we're talking about molecular genetics. But in your book, it's further over. So if you want these same experiments in your textbook, you have to look up these names I'm calling out. But the diagrams are the same. So if you look up Watson and Crick, it won't be in chapter 2. It'll be another chapter later on that you're actually going to be covering pretty soon. Any questions? No? Everybody's okay? DNA replications. There were 3 different ways that DNA could possibly replicate. Semi-conservative, conservative, or dispersive. And maybe you already know that DNA replicates semi-conservatively. It's not conservative replication. Conservative replication says that the 2 old strands separate, make 2 new strands, and then the old strands go back together. In semi-conservative replication, the original strands separate, but they never go back together. So they always have in DNA replication, you always have an old strand and a new strand. So DNA replication is semi-conservative. And dispersive, it says that this model will actually say that the DNA, the old strands will separate and literally break apart. So you have old and new components within a single strand. Of course, these were disproved by a person by the name of Meselson and Stahl. So dispersive was disproved, conservative was disproved, semi-conservative was proved. Meselson, say again.
Can you explain C again?
You can't see it very well here, but this strand is both strands are completely red. And they're red because they are labeled with radioactive nitrogen. But in the strands after it replicates, one strand is red and blue, just following one strand. It's no longer a mixture. And this one is still all red, and this one is all blue. This one is all red, and it's all blue. But in this model, you got an old strand that serves as a template for the new strand. So it's semi-conservative replication. Not conservative and not dispersive. And Meselson and Stahl did an experiment to show that it was not dispersive and not conservative. And they